Hello, everybody. I recognize a lot of some faces from pictures or real faces and a lot of names. So thanks for inviting me to talk to you guys tonight. Um, I went back and forth with a few different ideas of what to talk about, but I like instrumentation. So I'll probably talk a little bit about Mass Spec. All right, I did a lot of Mass Spec work in grad school. And I actually did pull up, not that I'm not going to go through it, but if any of you guys are interested in pursuing a graduate education, I do have my final thesis presentation. So I can bring that up and show you guys just a little rundown of what it looks like, and just give you an idea of what goes into graduate research, and the fact that maybe less than a third of it shows up in your final presentation. So it's a lot of hard work, and only a portion of it gets discussed. But I figure I'm going to talk about Mass Spec in forensic science. A lot of you guys, some of you I know are first year students, some of you are third year or fourth year students. So you may have gotten a description or explained how Mass Spec kind of works. But I like talking about Mass Spec mainly because it's a really versatile technique, and there's lots of different kind of pieces you can put together to make different instruments that do different things. So I will talk a little bit about instrumentation and the basics of it, but then I'll talk about some uses of Mass Spec in forensic science that you should all know at this point from going through 2216, and then go into a little bit more of like new research that's going in to Mass Spec in the field of forensic science. Not that any of it's really being used in labs right now, but it's kind of an emerging technique. Because some of it's really, really cool, and it kind of shows how versatile Mass Spec actually is. So I'll go ahead and share my screen. All right. I apologize for the very basic slides, but this is it. I think I used this presentation for another club on campus a few years ago. And it was in one of the rooms where the colors of the projector just went wonky or something. So I just had to do a very basic slideshow, but I'll try to make it interesting. So Mass Spec, you guys all should know this at this point in time. It's usually used as kind of a detector in some other techniques. So we pair it usually with things like GC. That's the most common technique that we use in forensic science. We can pair it with also LC, and I'll talk about chromatography in a little bit. So Mass Spec is used as a detector in certain systems. But it also has its own individual components, and it actually does have its own detector in it. So Mass Spec can actually be a detector inside of a detector. Watch Rick and Morty, it's basically like your simulation episode. But what does Mass Spec actually do? Mass Spec is a technique that separates certain components based on their mass to charge ratio. So that tells you two very important things that you need to know about Mass Spec. First, your things need to be charged. So they need to be ions, whether it's positive or negative ions, they have to be charged in order to see them using Mass Spec. And like the name implies, it separates these things based on their actual mass, so the size of these components. Again, you guys should know this, but Mass Spec is used to uniquely identify a compound. We commonly say that the chemical compound's mass spectrum is a fingerprint of the sample, much like our fingerprints as humans are unique to us. A chemical compound's mass spectrum is unique to it. You'll never have the exact same mass spectrum if you analyze two different chemical compounds. And there's a lot of information we can get from Mass Spec, and unfortunately in the field of forensic science, you don't really get a chance to look at all of this information. A lot of times in your instrumental classes, or even in your forensic science classes, we just say, hey, let's search this Mass Spec against a library. It comes up as a positive hit to cocaine, and you identify your sample as cocaine. There's a lot more information that can come out of a mass spectrum. Your molecular weight is one of them. You can also figure out elemental composition and even structural components based on all the little peaks that you see in a mass spectrum. Now, finally, it's great for forensic science because it's a very sensitive technique, right? It can detect things that are really, really low in concentration. And in forensic science, sometimes we only have a small amount of sample or very trace amounts of samples. So what actually does it do? There's three main components of a mass spectrometer. You have your ion source, your mass analyzer, and your detector. So you put your sample into your ion source, that goes through your ion source, generates your ions. Okay, it's the source of your ions, so it gives the things its charge. Those get sent into your mass analyzer, which analyzes those things based on their mass. And eventually they come through and hit the detector, which basically just records the signal, translate it into some language that the computer can interpret, which then plots your mass spectrum. Again, mass spectrum is going to be just a fingerprint of those chemical compounds. So your ion source, all it's going to do is make your ions. Now the ion source, it seems very, very simple, but the ion source is actually kind of the defining component of your mass spectrometer. And different ion sources will give you different mass spectra. So one thing I always like to point out when I talk about mass spec, especially in forensic science, because it's so common to just get a mass spectrum, search it against a library, or compare it to a known mass spectrum and say, yep, that's cocaine. Your ion source will dictate what your mass spectrum will look like. So if you analyze the exact same sample of cocaine using an electron ionization source or a chemical ionization source, your mass spectra will look different. You're going to say, well, I thought mass spectra were fingerprints of a chemical compound. And they are, but your ion source dictates what that fingerprint will look like. It's very important when we talk about mass spec and forensic science to understand if you use a different ion source, you're going to get a different mass spectrum. If you ever get a sample of anything or in a class, you're looking at online resources to find a mass spectrum of cocaine to compare a sample to, or anything that you got data for in a class to, you need to make sure you get the mass spectrum for the specific ion source that you used to analyze that sample. And now I won't go into the nitty gritty about how the chemistry works to ionize these samples. There's actually a lot of chemistry that goes into these ionization sources. But do know that these two sources that you see here, your electron ionization and chemical ionization, these are commonly used with gas techniques. So in gas chromatography, mass spec, you're going to have an electron ionization or a chemical ionization source. So if you're ever looking for mass spectra, again, online, most likely if you're doing GC mass spec, you're going to look for an EI or a CI mass spectrum. Now, there are a bunch of other types of techniques, ion sources. These ones are some of my favorite. Electrospray ionization and matrix-assisted laser desorption ionization, or MALDI. Electrospray ionization, it's pretty complicated, so we won't go into how it actually generates ions, but it's very soft. It's a soft ionization technique. What I mean by that, anytime you give any chemical species some charge, create an ion or generate an ion from it, there's a lot of energy that goes into that, okay? And what can happen if you input a lot of energy into something with bonds in it, those bonds can break. And that is called a hard ionization technique. Electrospray is a soft ionization technique. It's kind of like a gentle transfer of a proton or taking away an electron from your sample. And electrospray ionization is so soft. This is commonly not done, but they did it to prove a point. They analyzed an active virus using electrospray ionization mass spec. So they ran it through, they went ahead and generated a charge, they detected the ions, they got a mass spectrum, and they kind of rigged the instrument to collect the virus at the end of the analysis. Again, this is not something we commonly do. Mass spec is a destructive technique. Luckily, it doesn't use a lot of samples, so we don't really lose too much. But they collected the sample after it went through this instrument, and the virus was still active, right, after going through this process. So electrospray is really, really cool. Electrospray is also very commonly used for protein analysis. So those of you who may be interested in going to grad school and using mass spec, one of the more common fields that you'll use mass spec in outside of forensic science is proteomics. If you ever do any work with proteins, most likely you're going to be using an electrospray ionization mass spectrometer. Now MALDI is probably my favorite because it's one of the most common ones I used in grad school. Now MALDI is really, really cool. In this case, you mix your sample with another chemical that basically is really, really attracted to lasers, right? It takes the laser energy that you shoot at the sample, absorbs that laser energy, and then it transfers it to your analyte, and that's how you end up getting ionization. So not only does matrix-assisted laser desorption ionization use lasers, so you get to fire lasers at samples, it's also a very, very quick mass spec technique. Most of these other mass spec techniques, it takes a little while for you to get your sample ready to run it through the instrument. MALDI, you can get a mass spectrum in a fraction of a second. One other cool little side note about matrix-assisted laser desorption ionization, I keep saying it over and over because I love this technique, abbreviated MALDI, is much like mass spec is a detector inside of a detector. MALDI is actually an acronym inside of an acronym. Okay, so those of you who don't know, laser is an acronym. Laser is not, I mean, it's kind of a word now at this point, but laser is an acronym. It stands for light amplification through the stimulation of electromagnetic radiation, right? That's what a laser is. So MALDI is an acronym within an acronym. And because it's a mass spectrometer, it's a detector inside of a detector. So pretty cool. The other techniques that we can use for ionization are called ambient pressure techniques. Now, most of the time when we talk about ion generation, this has to occur at a very low pressure. Okay? It's really cool when we can start ionizing things at room pressure, right? In open air, right? It's very difficult to do, but we can do it. So I'll talk about that actually in a little bit. Once you generate your ions, it's going to go through then your mass analyzer. And your mass analyzer is a fancy way of separating different sized ions. One other thing I always talk about in mass spec, whenever you analyze a sample by mass spec, let's say again, we have cocaine. And those of you who know me, my favorite drug is cocaine. I know the mass spectrum of cocaine by heart. Don't ask me why, but I do. We're analyzing a sample of cocaine. We throw it through the instrument. There isn't just one molecule of cocaine in that sample. Remember back in your Gen Chem days, Avogadro's number, right? If you have a fraction of a mole of a sample, you're going to have a shit ton of molecules in that sample. All of these molecules of cocaine are going into the instrument. Some of them, maybe they stay intact, okay? They don't get a lot of energy, they get ionized, they stay intact, okay, and stay as the full molecule cocaine. Maybe some of them, they get bombarded with a bunch of electrons and part of the cocaine molecule breaks in half. So we get a different distribution of the cocaine molecule, whether it's fragments of it or a full cocaine molecule. So all of this stuff goes into the mass analyzer, and the mass analyzer separates it based on its mass. Now, the mass analyzers are really high-tech pieces of equipment in these instruments. They do some really cool things. They're more related to physics than chemistry, to be honest, and that's why you guys take physics. It's all kind of interrelated to one another, all these science techniques. But these mass analyzers, basically, it comes down to the fact that force equals mass times acceleration. Okay? We apply an external force to all these ions of different masses, and they are accelerated to different velocities. That's basically all these come down to. But there's a variety of different techniques that we can use. The easiest one to kind of describe, to give you an idea of how these mass analyzers work, is this top one, this time of flight. Where's my mouse? There it is. So the time of flight tube. Your time of flight tube is basically just a fancy race. At the start line, we give all of our ions the same energy, right? We kind of give them all the same amount of push. And eventually what's going to happen is, because kinetic energy is 1/2 mass times velocity squared, right? 1/2 mv squared. Your smaller mass ions will actually be accelerated to a higher velocity and will make it to the finish line first. Very simple. There's a lot of stuff that goes into it, a lot of voltages that are applied. So mass spectrometers can be very complicated instruments. When it comes down to it, they're very basic principles. Now, once these things go through the mass analyzer, again, smaller ones may come out first, followed by all your larger ones. As they come out, they hit the detector. And the detector is just kind of a fancy camera. Think about it that way. Now, because these are ions, we basically take these ions and they hit a chemical material that will emit a lot of electrons when something hits it. A lot of electrons get knocked off. And what happens is this amplification of signal, we take one ion and we get a bunch of negative charges from it, makes it very easy to detect when one ion comes out of our mass analyzer. This current basically is detected and it's transferred or converted to a signal. The instrument will kind of go through a high-tech calculation to figure out what mass to charge ratio that ion actually is. And then it collects all that information into your mass spectrum. And your mass spectrum basically gives you all of the ions that are detected and their mass to charge ratio. And also it gives the abundance of each of these ions. So how many basically counts, or how many times did this mass charge ratio ion hit your detector? And again, that produces a chemical fingerprint, because nothing is going to break like cocaine does, because cocaine has its own unique specific structure to it. Even if you have a chemical sample that has the same elemental composition as cocaine, the connections are different. When it breaks apart, it will produce a unique mass spectrum. Now, mass spec in forensic science is commonly not used on its own, right? It's used on its own in other fields, like in biology and chemistry. And that's kind of the cool thing about forensic science. Forensic science is it's an applied science. It doesn't necessarily have its own unique techniques, right, that somebody kind of came up with. Usually what it is it takes analytical techniques or methods from other fields of science like physics, chemistry, biology, and it applies them to something that was collected at a crime scene, right? So mass spec, it's used by itself in other areas, but in forensic science, it's commonly combined. My dogs might start playing, so if you hear any toys squeaking in the background, it's the dogs. Mass spec is commonly used in conjunction with other techniques. Mainly, this is chromatography, which is done before our mass spec analysis, or what we call tandem mass spec, which can be done after our mass spec analysis. Chromatography, like you all should know, separates components of a mixture. And because forensic science is a very dirty science, we never have pure samples, everything is contaminated or mixed with something else. Mass spec doesn't really work when we have a combination of things, all generating ions and all showing up in the same mass spectrum. So if we separate those components first, for example, a sample of cocaine that's cut with caffeine, we separate the cocaine and caffeine out, we analyze each of those separately by mass spec, and we can identify the components. We can also do tandem mass spec afterward, and I won't get into tandem mass spec because it's more of a, you got to get into the nitty gritty parts of mass spec to understand how tandem mass spec works. But if you are interested in it, come talk to me. You can find my email address on the website somewhere exists somewhere in the UNH website. And I'd be happy to talk to you more about mass spec. Now that we kind of understand how mass spec works, I want to talk about mass spec and forensic science, right? Again, mass spec is a very, very sensitive technique, and it identifies our sample. So it works really well with forensic science. We work with very small trace amounts of things. We do use separation techniques because, again, our samples are not pure in forensic science. So it works very well. And we all know, and you should all know, if you've taken 2216 with me or 404 with me, GC mass spec is the gold standard in forensic science, right? If you're unsure of how to analyze a sample, GC mass spec it. Most likely you're going to get something, right? Or if you're looking for an answer on a test if it's multiple choice, how do you analyze this sample? Most likely it's going to be GC mass spec. Now, in this case, we have a combination of two instruments that are hooked up together, and the mass spectrometer is actually acting as a detector for the gas chromatography technique. The only issue with gas chromatography is the fact that you need your sample to be volatile or be able to be converted to the gas phase for it to work. Most of the time for forensic science samples, we're working with very small molecular weight things, and this works rather well. If you try to analyze a protein, which is a very large biomolecule by GC mass spec, it doesn't work too well, or you've got to do a lot of sample prep to get the protein in a form where it can be converted to a gas stage. But luckily, most of our stuff works really well with GC mass spec. We all know that we can use it for drug analysis, right? In this case, this is the mass spectrum of cocaine, right? So we can uniquely identify any drug sample. We can run it through a library search. We can identify it. Also works great for arson and explosives. Arson, because most of our accelerants are very volatile chemicals, so they automatically already go into the gas phase. Very easy to analyze with GC mass spec. Explosives, sometimes people get a little worried with explosives with GC mass spec because in order for things to be converted to the gas phase, you have to heat it. And heat and explosives really don't mix too well. As long as you know the point where you can heat an explosive sample up to before it goes boom, you'll be okay. One of my grad students right now, he's finishing up a project where he does do explosives analysis on the GC mass spec. So as long as you're careful about it and you do your research beforehand, it's perfectly fine to do it. One other instrument we commonly see in some forensic science labs, usually larger forensic science labs, we don't really see this in small county kind of labs, is a liquid chromatography mass spectrometer, right? And in this case, liquid chromatography does the same thing as gas chromatography. It's just samples in the liquid phase. The liquid chromatography, if you're real worried about explosives, you can use LC Mass Spec to analyze your explosive samples. But this is commonly used in toxicology analysis. If we have biological fluids like blood or urine, right? They're already in the liquid phase. You run it through your liquid chromatography column. It separates out your components, and you can identify any kind of drug metabolites that are present. Oh gosh, my dog has got a toy. She's trying to find the squeaker. Now, it's kind of a newer technique in mass spec that's coming out. You may have heard this before. It's inductively coupled plasma mass spectrometry. In this case, there's no chromatography that's attached to it. This is a standalone instrument, but it's really cool because this actually gives you elemental information. And we know in certain cases, knowing trace elements can help identify a sample, right? It can make it more unique, help identify paint samples or glass samples. Now, ICP Mass Spec, I actually did a lot of work with ICP Mass Spec as well in grad school. It uses a fourth state of matter, right? So we have gas, liquid, solid. There's actually more states of matter than that. So somebody lied to you in high school. So plasma is actually a fourth state of matter. And the best way to describe plasma, it's almost like a jelly-like material. But it's a gas phase jelly material, if that kind of makes sense. Now, this plasma usually has a lot of radicals in it. And these radicals will actually result in the ionization of your sample. But because of this plasma and the nature of this plasma, again, I won't get into it. If you really want to know how this works, I'd be happy to walk you through how your ICP source works. It's actually pretty cool and pretty interesting. But because of how this works, you get elemental information. And this has actually been used to analyze paint samples. Okay, so this is paint samples using ICP, and actually looking at all the different layers in an automotive paint sample and identifying all these different elements that may be present. And it's also used to differentiate glass samples. And in the right-hand side here, this is actually a principal component analysis, or PCA analysis, of a bunch of different glass samples, 4 of them. And what you can see is the groupings that exist, right? There are some outliers in certain areas, but basically what this PCA analysis tells you is that we can differentiate these 4 glass samples using elemental information. Those are the more common ones you may see, at least with mass spec analysis. With ICP mass spec, you can also add what's called a laser ablation source to it. We love our lasers in mass spec. And this LAICP mass spec can actually be used for mass spec imaging, which I'll talk about in a second. But these are the more common ones that you may hear of or see in forensic science. What I really wanted to talk about is kind of the newer techniques that you may not know of that are actually doing some really cool or interesting research. So one of these is called Desorption Electrospray Ionization, or DESI, DESI. Very, very similar to electrospray ionization, where we're going to spray some solvent that has our sample in it, goes into the inlet of the mass spectrometer, and we run our sample and get our analysis. DESI no, Bailey, no, not that toy, please. That's the loudest one that we have. Thank you. They get excited when I get home and all they want to do is just play. So DESI, it works very similar to electrospray, where we do have a solvent spray, but that solvent spray, we actually shoot it at a surface. Okay, so we actually have a sample on a surface, whether it's a glass slide or if it's on a toothpick, right? It's a toothpick saturated with solution, and we spray our solvent at it. Now, this action, what happens is the liquid solvent will dissolve some of the sample on the surface and will kind of take it off with it and go into the vacuum of the mass spectrometer. Okay, so the vacuum kind of sucks the liquid up with whatever sample's in there. And DESI is really, really cool because we can literally hold objects up to this instrument and analyze it. So there really isn't sample prep. We can go ahead and hold the sample right up to it as it is, and then take that sample away from the mass spectrometer and then still maintain it and keep it for further analyses. So DESI has been used for drug analysis. In this case, this is a cocaine sample or multiple cocaine samples that were identified as cocaine, but then also we used it to identify impurities that were in the sample as well. And again, this literally was just taking the powdered sample of cocaine and holding it or putting it on a surface where that spray came in contact with it. We've also used this for explosives and also chemical warfare agents. In this case, we use it to identify a mixture of explosives. I think in this case, what happened here was they had to, because the explosives were such a small, fine powder, they had to stabilize it on a surface with some cream. So they put cream on it, put the powder on top of it, and they analyzed this with DESI. And they were able to identify all of these agents. And we also used it for chemical warfare agents. This becomes really, really cool because now we have what we call a standoff technique, where if we do have dangerous or hazardous chemical compounds like explosives or chemical warfare agents, we don't need to further prep the sample. And sample prep sometimes can release more volatile chemicals or can make your samples more hazardous and more reactive. In this case, we just hold it up to the instrument. When we're done, we put it back in its nice, safe container, and that's it. So there's minimal exposure from the analyst and minimal possibilities that you're going to get hurt. Now, DESI can also be used for imaging analysis, and this is where Mass Spec becomes really, really cool. I did a lot of work with imaging Mass Spec in grad school, and the reason why we like imaging so much, at least in a forensic science standpoint, is because if you're trying to demonstrate something to a jury, pictures like this are better than complicated mass spectra, right? A jury's not going to understand what the hell all these lines mean. But if I tell you right now that sample right there, all the red that you can detect in that fingerprint, all the ridges that are there, actually cocaine, okay? What that tells you is that this person handled cocaine at some point, and we can detect that in their fingerprint ridges. Okay, the cocaine basically stayed in their ridges, and we can detect it in mass spec, right? It's a lot better than just showing you the mass spectrum of cocaine and saying, hey, I pulled this off of somebody's fingerprint, right? This has a lot more of an impact than your mass spectrum. This image on the left here as well also has a great impact. In this case, we have somebody wrote 2700 with 2 different red pens. This wasn't an actual case, it was just a research study. But they wrote 2700 with 2 different red pens. Pretty much look identical, right, to the naked eye. There's not much different there. They both look like red ink. When you analyze them using mass spec, what you can see is that both inks have this same ion present, ion at 415. When we start looking at different ions, what we noticed was in the 27, there's an ion that exists at 399 that is not present in the 0 ink. Right? That kind of shows that there's 2 different chemical components present, and we can use it to distinctively show these are 2 different inks, right? Anybody looking at this photo, if you give them a brief explanation of what it shows, they'll say, oh yeah, that makes sense. The 0's disappeared because it's not the same ink as the 27. Now, how these imaging techniques work, actually, no, I think there's a slide that I do have a slide that talks about mass spec imaging. And actually, I don't know if the chat is for me or anything, but okay, cocaine changed your finger. Okay. I was like, I saw the little 3 at the top, and I'm like, oh shit, I should have the chat open as well. So yeah, so cocaine, you can actually, and people have done studies like this with fingerprints with not drug samples, but even just lotion, right? My hands get so damn dry in the winter. I've got to put lotion on like every single day, at least 3 times a day. If I went ahead and touched a surface and we analyzed it by mass spec, we may be able to identify my fingerprint because my fingerprint will have specific ions in it from my lotion that yours may not have if you use a different lotion or don't use lotion at all. Yeah, don't try to burn your fingerprint off. It might not feel good. So I'll get into how imaging mass spec works in a second. It's very complicated. To get an image like this for this fingerprint, there's probably, looking at the level of detail in this, there's probably about 10,000 to 100,000 mass spectra that were used to generate this image. So my data, I have a laptop just just dedicated to grad school because my analysis and my result files are massive. I'd have like an imaging run that would take like a terabyte of data. Right? It's insane because there's a lot of information here, but I'll explain it in a second. Another cool mass spec technique is DART. This is direct analysis in real time. And much like DESI, you literally just hold something up to the inlet of your mass spectrometer, it sucks some of your sample in, and you get a mass spectrum. In this case, there's no solvent. You're not spraying it with anything. It's literally just a combination of heat and the vacuum from the mass spectrometer that will draw your sample in. Now, in this case, this really only works for very low molecular weight things, less than about 800 Daltons. But again, most of the stuff we're analyzing in forensic science fall under this category, right? For example, cocaine, molecular weight is 303, right? So we'll be able to see it with this. And people have done a lot of studies with DART, again, for things like chemical warfare agents and explosives, again, very hazardous materials. Drugs is probably one of the most common things that we'll see with DART. They've even done it with ignitable liquid samples. So they have something soaked in some ignitable liquid, they hold it up to it, the ignitable liquid comes off the sample, goes right in. They've also even done this with contaminated food, right? Not necessarily forensic science. For example, they've been able to detect pesticides on specific vegetables or fruit products using DART. Now, in this case here, this sample that you see, this is actually an herbal sample, and somebody went ahead and doped synthetic cannabinoids onto this herbal sample. All you do is you literally hold up the herb to the inlet of this mass spectrometer, and what they were able to do is they were able to identify these synthetic cannabinoids. Okay? Really cool. Takes about a couple of minutes for you to get a mass spectrum. We can also use it to directly sample either biological fluids or pills, okay, or tablets or powders. And in this case, what they looked at was they had a pill that somebody was marketing, I forget, as a specific drug. I don't remember exactly what it was. And what they saw was when they held it up to the mass spectrometer, they didn't get components of whatever this drug was supposed to be. When they compared it to a real sample of this drug, they realized that somebody was making counterfeit medication and selling it to individuals. And again, this entire analysis probably took about 10 minutes total time. Again, we can use it with explosives analysis. And again, this is it's a good thing to have for hazardous substances, again, because you don't want to be exposed for too long with these hazardous substances or things that can go boom if you're not careful. Now, this ambient ionization technique of holding something up and not having it be in a vacuum chamber, really, really cool, very useful for forensic science, especially if we can take that and turn it into a portable instrument, right? Imagine taking or walking up with a little handheld mass spectrometer, and you literally just take your sample and hold it up to, and it says, oh, this is cocaine, you should probably collect it. You go ahead and collect that unknown white powder now, right? Instead of just assuming that it could be a drug, maybe doing a color test to see if it's presumptively cocaine, then getting it back to the lab and realizing it's only caffeine, right? So this can be really, really cool. And a lot of people think that this ambient ionization technique is new, but actually it was originated in 1972. Okay, and this ambient ionization technique was actually used to detect alcohol coming through people's skin. And I think this is amazing, right? And it's also pretty funny. Literally, they rigged up an instrument. Okay? It was hooked to a mass spectrometer. And what they did is they had a chamber, and somebody would stick their arm in the chamber, hold it there for a certain amount of time, and as the alcohol permeated through their skin via sweat, it would go into the mass spectrometer and they would identify it, right? This is the first ambient ionization, right? You're sampling something at ambient pressure. This is the first ambient ionization mass spec technique, which I think is really, really neat. I didn't show an example spectrum. This is a note, I guess, again, from a previous presentation. Now, one of the things I talked about is making things portable, right? That's the newest kind of research area for forensic science, is taking some of these lab-based techniques, these instrumentation techniques, and bringing it to the scene, rather than waiting for the sample to be packaged, processed, looked at in the lab, sent to an instrumental lab and run through some instrumentation, right, that may be backed up with lots of samples. taking those instruments to the crime scene is the newest kind of area of research. And there are a few portable mass spectrometers that are out nowadays. We actually have one in the forensic science department, the portable GC Mass Spec. The biggest issue with portable instrumentation is it's never as accurate and it's never as sensitive as your lab or benchtop based techniques. But again, it does give you a level of confirmation that something may be present at the crime scene. One of the first instances of a portable mass spectrometer, they actually had this instrument here, okay? It wasn't necessarily like a handheld machine. In this case, you'd put it in like the seat of a car, and that's actually how they first started this. And literally, this was just a lab that decided to home build a mass spectrometer and try to make it as small as possible. But what they did is they used this in a car, and they drove this around an area where a clandestine lab was suspected of being. Okay, so this building here in this area, it was suspected of being a clandestine lab where they were making methamphetamine. Without going into the lab, all they did was they drove around it multiple times, and the mass spectrometer took little samples of the air, ambient air, analyzed the air, and what they found was they found precursor chemicals that were in the air that were used to make methamphetamine. And these little dots down here basically tell you how intense, or basically the count or abundance of each of these ions that were detected. Okay. Imagine doing this. Imagine not having to necessarily get a search warrant, right? To go ahead and look around a clandestine lab, right? Instead of going and knocking on the door and be like, hey, how's that meth coming? You can go ahead and drive around, sample the air and say, you know what, Judge, look, there's proof here that we've got methamphetamine being synthesized. We need to go and bust them, right? Because the air, you can sample the air, right? They don't own the air. So that was actually kind of one of the first. Chat keeps disappearing. Oh my gosh. That would be, oh my God, could you imagine me running around all the dorms and being like, oh, who's got weed? Running and sampling the air. Oh my goodness. So the airspace, it is to an extent somewhat their private property, but they can't really control it. All you'd have to do is just sample it slightly further away, and you can still see they were detecting some of these chemicals even down further on the main road, right? I don't know why every time I click, the chat goes away. I'm gonna try to keep this here. So this is kind of one of the first portable mass spectrometers, or the instances of them using it. Again, it was a home-built instrument, but nowadays we have actual commercial portable mass spectrometers. These are both commercial portable GC mass specs. This green one here, we have this in the lab. Okay, if you ever in, was it room 414, the instrumentation lab on the forensic science floor, you can walk through the benches and you'll see this puppy sitting right on the bench, right? This one is an even smaller version, okay? So this is what we're trying to achieve now, right? With mass spec, or even with any kind of instrumentation, we're trying to make it small, we can carry it to the scene. One other cool area of mass spec, like I've already talked about, is mass spec imaging. And I think imaging is really, really cool because it gives you a really neat demonstrative kind of capability in a courtroom. Now, how mass spec imaging works very well with laser-based techniques, okay, or desorption electrospray ionization. It works well with things where you can ionize or sample a very small area, then move over and sample another small area. So what happens is, it's easiest to describe this with a laser, right? Imagine a laser pointer. You can go ahead and point the laser pointer at specific dots on a sample. If you set up these dots to be a grid almost, right, a 100 by 20 grid of all these dots, what's going to happen is as you sample one dot, all that information gets sent to the instrument, you get a mass spectrum for that dot. You move to the next one, you get another mass spectrum. You can do this over an entire surface area. Now, these take a little bit more time to do. I would do imaging experiments on mouse organs. We did a study with nanoparticles in kind of a drug delivery aspect. And I would image organs, so little tiny mice organs. So they're not very big. They were probably about as big as like your pinky nail. and we'd go ahead and image them, and it would take, depending on the resolution, so how many spots we had all over the sample, sometimes it would take about 12 hours to do. So it does take a very long time, but it gives you a lot of information, because then what you're going to do is you're going to take all of those mass spectra and you're going to say, I know this specific ion for that fingerprint is related to cocaine. And it's going to generate an image that shows you exactly where that ion is detected, right? So in this case, we actually show these are organs, and this is very commonly done in the biochemistry field. But what you can see here is all these different ions. For example, in this image down here, we took the same slice of whatever organ this is, and we detected different ions. The green one shows up in different areas than the purple and the red. And this can give you information about what may be detected in specific parts of this image. Now, how can this be used in forensic science? There's a lot of research going on with this. And again, I think it's really, really neat. One of the cool ones is hair analysis to look for drugs, right? You know me, I love my drugs. And what happened is they looked at your hair sample. Remember, your hair is like a little diary of your drug use, right? Hair grows about 1 centimeter a month. Drugs will get incorporated in hair as it grows. Your hair is dead, right? Your hair is actually not living, okay? So as soon as something gets incorporated in it, it stays, right? So what happened here is they went ahead and ran 4 different samples of hair, okay? So the first one is unwashed hair. Now, unwashed hair, the issue with unwashed hair is you could have environmental exposure to this drug, and it could stick to your hair. So that's what we end up seeing here. All this heat map here, all the red, is actually an ion indicative of cocaine. Now we went ahead and we washed it with water for 20 seconds. Really doesn't help. Now what we end up seeing is as we bleach it, we continue to see a decrease in cocaine, but we can still detect it, right? Now the process of bleaching, if you know anything about hair, it can remove some of the drugs. In this case, I believe these hair samples were not actual cocaine users. They mimicked cocaine use, and they wanted to see if bleaching could remove it. Now, in this case, this next study, though, lights up like a Christmas tree, right? In this case, we have, how many is this? 5 hair samples. We have a control hair sample on top, no cocaine at all. We have 2 hairs here of cocaine users, avid cocaine users, and these 2 at the bottom were non-cocaine users, okay? These 2 hairs of cocaine users lit up like a Christmas tree with Mass Spec imaging. And again, this is really neat to show in a courtroom setting, right? It shows that these samples, we detected cocaine in these hair samples, and it shows that these are very avid cocaine users because it's continuous throughout the hair. If there was only one little tiny band of cocaine, though, remember, we could go ahead and if we design our experiment to raster and sample specific portions of the hair, depending on which portion we analyze it or detect it in, we can figure out that somebody did cocaine 3 months ago or 8 months ago, depending on where it is. Another cool example is fingerprint analysis, not just for detecting cocaine. This is actually talking about detecting specific lotion ions that are detected or different things like, again, there could be cocaine or THC in a fingerprint. But the cool thing about this is, remember, fingerprints can be found on anything, right? And sometimes they're found on a background image that's very messy or noisy. It can be hard to see the detail in the fingerprint. We analyze it by Mass Spec. We can actually eliminate all of the background. We get a nice clear fingerprint. Did you raise your hand?
I did. I have a question.
Yes.
I don't know if this is a dumb question, but you know those CBD lotions?
Yep.
So what would like pop up on that scan?
So it depends on the amount of the THC content. So CBD creams, the reason they're different than marijuana is CBD has a much lower THC content. It's a low enough THC content to where if you go and lick the cream, you're not going to get high, right? Just like with CBD oils, or they even have CBD cigarettes. There's not enough THC in there to induce an effect.
Oh, okay.
Depending on the mass spec technique and its sensitivity, you could still detect THC. right? If it goes down, your detection limit is low enough to detect the small amount of THC in those CBD creams or whatever, you still may see that ion, right? Now, the interesting thing with that, because marijuana is not legal in every single state right now, you may have to quantify the amount of THC to prove that it's actually CBD and not actual marijuana sample.
Right, yeah.
It's really, in Mass Spec, great for quantitation. Mass Spec is an amazing tool for quantitation if you do it right. So we can image fingerprints, but one other cool thing that we can do, it's like, but wait, there's more, right? For $9.99, you can get, what we can do is we can separate fingerprints. If my fingerprint is different than yours, if I use a different lotion than you, and our fingerprints are overlapping, we can use Mass Spec imaging to actually deduce detail where they're overlapped that pertain to each of these individual fingerprints. Imagine pulling this at a crime scene. You have overlapping fingerprints. You have no idea what ridgeline belongs to what in that center area. You can use Mass Spec imaging and find unique ions for each of the fingerprints, the left and the right, and then separate it using this imaging technique. Because for image B versus C, you're showing which specific ions come up, which I think is really, really cool. We also do this, like I said, to deduce fingerprints that are on a very noisy or difficult background. In this case, this is a fingerprint on a cigarette container over the barcode. And you can see that the barcode is obscuring some detail, right? Black and white, it's very difficult to find a fingerprint powder or something that you can use to basically enhance this fingerprint without losing information. Mass Spec imaging, what we can do is we figure out ions that are unique to the ink versus the fingerprint itself, and we can separate the images. Now, a few other things. I think there's only a few more slides, so bear with me. I know it's, I don't know how long your meeting lasted, but I'm assuming it's about an hour. So I will finish by 7 o'clock, I promise. DNA, we can actually get a DNA profile using MALDI, Mass Spec. Now, it's very different, okay, the type of DNA profile that you actually get when you do DNA. In this case, when we do DNA, remember we're looking for short tandem repeats, but we can actually use MALDI to sequence a DNA sample. We can figure out the exact nucleic acids that are present in that DNA sample, right? It's also, because it's MALDI, remember MALDI takes maybe a fraction of a second, maybe a few minutes to get a sample or get a spectrum. Very, very quick. Now, there still is sample prep. You've still got to do your PCR. You still got to do some sample prep before you throw it in the instrument. But the analysis, the instrumentation analysis between MALDI versus capillary electrophoresis, which is usually what we use for DNA analysis, MALDI is much, much quicker. So not only is Mass Spec useful in the field of forensic chemistry, it's also crossing over into the field of biology. And actually, I'm doing a project, well, my grad student is working, my new grad student's working on a project that an old grad student worked on that I got a paper published about, is actually using GC Mass Spec to analyze hair samples. And what we're doing is, usually you can't get a nucleic acid DNA or nuclear DNA sample from hair, right? Unless you have that follicular tag attached. So what happens is we either do mitochondrial DNA, which is still not unique, right? It's not individualizing. Mitochondrial DNA is also very, very time consuming and expensive. So you can only do it at specific labs. You have to send the sample out if they approve mitochondrial DNA to be done on the sample. But you still may not get individualizing information. So what I'm working on is using GC Mass Spec to analyze the proteins present in your hair, okay? And looking at that as a way to individualize hair samples. So my grad student and I, we both have very similar hair. We looked at it under the microscope, very, very similar morphological characteristics. So if we can't do DNA, an analyst may say they're indistinguishable, right? I can't tell if they come from different people or the same person. Rather than that being it, you send this hair sample to be analyzed by GC Mass Spec. And because proteins are directly related to DNA, we can still get unique information from it, right? Remember your central dogma of biology? DNA goes to RNA, goes to proteins, right? If you get a little mishap in your DNA, right? And how DNA works, you get chunks that code for specific proteins. Everybody that's a human has a portion of their DNA that codes for hemoglobin. Some people, there's a little mutation that occurs in their DNA. And the hemoglobin, it's God knows how many amino acids long, one of them is not correct. In some cases, this doesn't really cause a big problem, right? Hemoglobin still functions as it's supposed to. In some cases, it can cause a disease, like sickle cell anemia is caused by a one little amino acid mutation in the protein hemoglobin. If we can detect that, we may be able to differentiate my hair proteins from your hair proteins. And we actually did prove, we looked at the amino acid amounts basically in our hair and proved that they're different between the two samples. So rather than the analyst saying these two hair samples are not consistent or we can't tell the difference, you move to hair protein analysis or amino acid analysis, and you could say, well, actually they are different. So they're not from the same source. So Mass Spec, it's got a wide range of uses. So this is just kind of a summary about Mass Spec. But there's a wide range of uses, and honestly, Mass Spec is probably one of the most common instruments, at least in the forensic chemistry side, that's being currently researched, right? A lot of people do forensic Mass Spec research because it's unique, right? It's sensitive, and it's a great overall tool. Now, those of you who may end up going into grad school, if you are interested in instrumentation, Mass Spec is the way to go. If you're ever looking at a project to do, use Mass Spec if you're in the chemistry world. And honestly, even if you're in the biology world, if you do protein analysis with Mass Spec, you're still going to be pretty good, right? It's going to give you that instrumental experience that labs will be very appreciative of. I think that's it. I know I said, I have my